Every living black-footed ferret on Earth descends from just 7 animals, a genetic bottleneck that left an entire species one outbreak from collapse. Then, in 2020, scientists took cells frozen in 1988 from a ferret named Willa, an animal that never once reproduced, and cloned her. By 2024, she had effectively become the 8th founder. But understanding what that means starts with why the prairies themselves began to disappear. Across the Great Plains, black-footed ferrets once roamed a grassland expanse stretching over 1.5 million square kilometers. Their survival was inseparable from the prairie dogs that dotted this vast region. Prairie dogs supplied almost all of a ferret's diet and provided the intricate burrow systems needed for shelter and raising young. When these colonies thrived, so did the ferrets. But as the 20th century advanced, the relationship between people and prairie dogs shifted. Ranchers and government agencies saw prairie dogs as pests competing with livestock for forage. By the 1970s, coordinated poisoning campaigns swept across the West, removing more than 90% of prairie dog colonies from the landscape. This was not a slow decline. The loss of prairie dogs meant the loss of both food and shelter for black-footed ferrets, collapsing their world almost overnight. Even in the few places where prairie dogs survived, disease struck hard. Sylvatic plague and canine distemper swept through the remaining colonies, erasing what little resilience the ecosystem had left. By the end of the 1970s, the black-footed ferret's range had shrunk to a handful of isolated pockets. The species was not just losing individuals, it was losing the entire system it depended on. The collapse of the prairie dog network set the stage for one of the most dramatic conservation challenges in North American history. At 3 in the morning on September 26, 1981, the quiet of a Wyoming ranch was broken by the frantic barking of a dog named Shep. John and Lucille Hogg found Shep outside their porch with a small, unfamiliar carcass. In the dim light, the animal's black feet and masked face revealed what experts had long feared was gone, a black-footed ferret. The Hoggs contacted wildlife authorities, and within weeks, federal biologists arrived in Meeteetse. On October 29, 1981, U.S. Fish and Wildlife Service biologists captured a live male ferret from a nearby prairie dog colony, the first confirmed wild capture since 1973. The animal was fitted with a tiny radio collar, its movements tracked in hopes of finding others. News of the discovery spread quickly. On November 6, 1981, the U.S. Fish and Wildlife Service issued a press release confirming that black-footed ferrets still survived in the wild. The agency launched an emergency response, mobilizing teams to survey the landscape, assess the population, and protect the fragile colony. For the first time in years, the prospect of recovery seemed possible. The Hogg family, through a single night's vigilance and a ranch dog's instincts, had reopened the story of a species that science had declared lost. The Meeteetse colony became ground zero for a new chapter in North American conservation, as federal and state partners raced to prevent a second and final disappearance. Between 1985 and 1987, wildlife biologists captured 18 wild black-footed ferrets from the Meeteetse colony in Wyoming. This group represented the last hope for the species, but the numbers alone hid a deeper problem. Out of all 18, only 7 individuals produced offspring that survived and bred in captivity. Every black-footed ferret living in the recovery program before 2020 descended from these 7 founders. The genetic bottleneck that resulted was severe. With so few ancestors, the population carried only a fraction of the genetic diversity once present in the wild. This narrow family tree meant that the species was vulnerable to inherited diseases, reduced fertility, and an inability to adapt to new threats. Conservation biologists recognized that while the captive breeding program had saved the species from demographic extinction, it had also locked it into a fragile genetic corner. The search for a solution became urgent. To secure the species' future, scientists needed not just more ferrets, but more genetic material, an infusion of diversity that the existing 7 founder line could not provide. Willa, a wild-caught female black-footed ferret, died in January 1988 without producing a single offspring. At a time when every living ferret traced its ancestry to just 7 founders, her genetic line seemed lost forever. Yet, before her death, a small sample of skin cells was sent to the San Diego Zoo Wildlife Alliance's Frozen Zoo. There, under the direction of conservation geneticist Oliver Ryder, Willa's cells joined thousands of other endangered species tissues, cryopreserved at -190 degrees Celsius. The sample was meticulously cataloged, stored in liquid nitrogen, and tracked through decades of technological change and staff turnover. For 32 years, Willa's genome remained viable but untouched, a genetic insurance policy with no clear path to use. In December 2020, that changed. Scientists used somatic cell nuclear transfer, a technique that replaces the nucleus of a domestic ferret egg with the DNA from Willa's frozen fibroblasts. The resulting embryo was implanted into a domestic ferret surrogate. On December 10th, Elizabeth Ann was born, the first cloned endangered species in the United States, and the first black-footed ferret in over 3 decades to carry Willa's unique genetic legacy. This single birth demonstrated that cryopreserved cells could restore genetic diversity erased by history, and that a lost founder could return to the living population. For conservation biologists, Elizabeth Ann's arrival was more than a technical milestone. It was proof that genetic rescue, once theoretical, could become a practical tool for saving species on the edge. Noreen and Antonia, both born in 2023 from Willa's preserved cell line, moved the story of black-footed ferret cloning from isolated achievement to active conservation tool. Their arrival meant that Willa's genetic material, once locked away in a frozen vial, was now present in living, breeding animals. In June 2024, Antonia gave birth to 2 healthy kits, Cybert and Red Cloud, at the Smithsonian's National Zoo and Conservation Biology Institute. This event was more than symbolic. Genetic analysis confirmed that these kits carried alleles not seen since the 1980s, restoring lost genetic variation to the entire captive population. Within 5 years of Elizabeth Ann's birth, 15 ferrets in the breeding program carried Willa's unique genetic material. For the first time since the 7-founder bottleneck, the recovery program's family tree broadened. Willa, who had died without leaving any descendants, was now recognized as the species' 8th founder. Conservation biologists tracking heterozygosity saw a measurable increase, about 3%, across the captive population. The introduction of Willa's genome offered a concrete answer to the challenge of inbreeding, demonstrating that cloning could deliver real genetic rescue. What began as a frozen insurance policy had become a living, multiplying contribution to the survival of the species. Ryan Phelan, executive director of Revive and Restore, described the arrival of Elizabeth Ann as the beginning of a new era for conservation, one where lost genetic diversity can be revived and put to work for endangered species. Yet the optimism of this breakthrough stands beside a series of profound ethical and practical questions. Cloning requires the use of surrogate mothers, often domestic ferrets, raising concerns about animal welfare and the risks involved in repeated embryo transfers. The cost of advanced biotechnology can also divert resources from habitat restoration and field-based recovery, which remain the foundation for any species' survival. Conservation biologists debate whether introducing genetic material from decades-old cell lines could bring unintended ecological consequences, such as genetic incompatibility or disease risk. Oversight of frozen genetic resources remains unsettled, including who owns them, who decides their use, and how benefits are shared. As the black-footed ferret becomes a test case, the world is watching to see how science, ethics, and policy will shape the future of genetic rescue. Sylvatic plague remains the most persistent threat to black-footed ferrets and the prairie dog colonies they depend on. Even as genetic rescue expands the species' gene pool, disease outbreaks can erase years of recovery in a single season. Conservation biologists are now applying the same biotechnology logic behind cloning to breed ferrets with natural or engineered resistance to plague. This includes selective breeding for immune traits and experimental gene editing to introduce protective alleles, efforts that, if successful, could reduce the need for chemical treatments and mass vaccination campaigns. Meanwhile, the concept of biobanking has moved from an isolated experiment to a national policy priority. The San Diego Zoo's Frozen Zoo and its partners are working to collect and freeze tissue samples from every endangered species in the United States, treating these archives as genetic insurance against future losses. For black-footed ferrets, the cloned lineage is not a replacement for habitat restoration, but it now continues through the breeding program, with Willa's descendants projected to play a role in recovery efforts through 2025 and beyond. Today, black-footed ferrets carry genetic material from an eighth founder, willa, altering the trajectory of a species once locked in a perilous bottleneck. As biodiversity crises accelerate, experts argue that precision cloning could become a lifeline for other species on the brink. Biotechnology now shapes the boundaries of extinction and survival.